Your reality is my reality.
That's what you're
All right, Jill. Let's pray, and we'll talk about the end of the year, and we'll review for a test, and just a little bit, not the whole day, and then we'll do some DNA. Okay, let's do that. Let's pray. Father, thank you for this morning. Thank you for the rain that we've had. Thank you for the sun that you'll bring. Thank you for the food that we've eaten, and just everything that you've provided for us. Lord, as we're here today and we look one more time at photosynthesis and respiration, may we see just truly the miraculous in these things. That they're so intricately and perfectly ordered. Only you can do this. Father, as we look then to DNA and see something of your master plan and how you've written all this and literally into your creation, may we be in awe of you and seek to know you. We pray this in Jesus' name. Mm-hmm. All right, so the end of the year. Goodness. Get out your calendars. Let's look at that. Okay.
Why did I pull up the calculator?
Y'all there? What's the date for Thanksgiving?
The 28th.
Thursday the 28th. Are we supposed to have class the 26th? Is that right?
I thought we were off that whole week.
No, we could add on the
The
22nd. The 21st, yeah, we could add on the 21st.
I'm hearing all kinds of different.
Because I had asked, what's his name? Mr. Eccles yesterday.
Yeah.
And he said that our last time being physically in class is the 21st. And then we're off Thanksgiving, and then we have finals that first week of December.
Okay, so my Canton kids that are worried about this, they don't even need to worry about it.
So do we come back during December?
No.
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 We don't
You'll
come back to
take
January.
all of all final exams are online. So I will let you know what that's going to look like for this class. I have a chemistry class that I'm doing online, so at least my head's a little bit in all this. You will have a written final exam. You are going to, when it comes time for this, I'll give you 2 hours to do this, okay? You're going to need to print, my recommendation would be to print your final exam. So, like, get yourself in a good way, okay? Print your final exam, write on it, take a picture of it with your phone, and email the pictures to me. Then I'll get it, I'll print those pictures, and I'll grade your exam. Okay. I'm not worried about all this with the, they've got different things where you can't cheat or what. Nonsense. If you want to cheat, you're going to cheat. So it is what it is. I hope you don't.
Sorry.
Not encouraging when you laugh. But
Sorry, no.
maybe I'll just make it an hour. I had Dr. Arison as a professor, and I had several classes with him that were online. And one of the things he said was, well, I just make it so tight. Like, yeah, it's open book, it's open notes, open whatever. But I'm going to put a tight time limit on it, so that if you don't really know your stuff, what good's it going to do you? But at any rate, it's going to be what it's going to be. It works out real well in chemistry because, it just takes a while to do the math. In this, I guess you could Google stuff, but, we'll see what it is. So that's going to be the deal. So just a reminder, you need, because there will, the time you open the test, I get a timestamp. And the time you send me your email, I have a timestamp. You have 2 hours to open it, print it, do it, email it. Okay? It's going to be plenty of time. That's giving you extra time for technical glitches and stuff like that. You know what I mean? Obviously, if there was some immediate problem, like you opened it but you didn't see anything or whatever, then you're going to email me immediately. Okay? That's not going to happen. I haven't had any problems with this with anybody. So, all that's going to work out. But I don't know how your other tests are going to be online for your other classes, but in this class, that's what that's going to look like. But all of your final exams will be at home, will be online. Okay? I'll finish your breakfast quickly. We got to roll. Okay, so the BMA, you're at a BMA school. Do you even know what that means? Those initials? It's Baptist Missionary Association. They broke off from the Southern Baptist Convention in like 19 something, early first quarter of the last century. It's weird to say last century, I was born in that one. I know you aren't, but anyway, I'm old. So, they're big on missions. You don't need the whole history, but you need to know that this association, there's a national organization. They actually run the seminary up the ways. Then each state has their own state association. Well, it's the Texas State Association that in 19 whatever decided to found a college here in Jacksonville. And so it's the State Association of Texas that runs this and largely funds this school and things like that. They have a meeting every single year for all the churches, and this year it's in Jacksonville. So they're holding it at 3 locations. They're holding it at First Baptist Jacksonville, at Cornerstone Baptist Jacksonville, and on campus here. Your professor is a pastor in the BMA of Texas, so I would be at this no matter what. It just so happens that things are happening here and will be on campus, and so the afternoon classes are canceled for Monday and Tuesday, the 18th and 19th. Okay. You have a morning class with me, but you have a morning class on Tuesday. I've got a lot of stuff happening in the morning on Tuesday. I will not be here. Okay. So our class is canceled for November 19th.
So I won't see you at all after that.
No, you'll see me on Thursday.
Okay.
Yeah.
Okay.
I'll be here Thursday. You'll be here Thursday. Okay.
Hopefully.
Hopefully. What else are you gonna do? You live in Tyler. What
No,
else?
I live that way. My great grandma was in Tyler.
Oh.
She went to the doctor in Tyler, but I live in I live in Dew. I live in Fairfield.
I live in Fairfield.
You lived in Fairfield?
Yeah, where you going to church in Fairfield?
Round Prairie,
Okay.
Chris Storage,
Sometime come check out Calvary.
Calvary.
Yeah. You don't even know.
We went there when I was little, but it got bad.
Well, I guarantee you whatever it was, it is not now.
Yeah.
Guaranteed. I'm going to pray that your car breaks down right in front of there on a Sunday morning and you have no choice but to come in. You're going to need somebody to fix it.
I'm fine.
Just kidding. But Aaron, I am the new pastor at Calvary.
In Fairfield?
In Fairfield.
They're both the same.
Really?
So I don't know which is
Yeah.
the side of the see which is which.
They voted unanimously to select me last Sunday morning, and we went out Sunday night to be installed.
Really? Like Calvary by the post office in Fairfield.
I don't know the post office. It's catty corner from the county
Yeah.
courthouse.
Huh. Do you live in Fairfield?
Lord willing, I will be within the year. I don't mean by December. That's way too soon.
Don't live in bad places, Fairfield. I live like 20 minutes from Fairfield, but I say I live in Fairfield.
Hopefully, we will actually live in Fairfield. Because, yeah. Anyway, now you all know that you didn't really need to know, but that was fascinating. She just trashed my church, but it's okay. It's the Lord's
Well, it was
church.
bad when somebody else was there.
Well, I don't know about all that. You should, maybe just one time you go, oh, Dougie's over there. You don't call me Dougie. When I taught forever, everybody called me Dougie. So I'm just so used to that. Right?
They called you what?
Dougie.
Sorry. I'm not
I started teaching when there was that song, Can You Teach Me How to Dougie? And I got that all day, every day. Are we ever going to learn any science? Just random things about me.
I think we should be just over this conversation. I think scrap
What
biology.
was your nickname when you taught high school?
And you start saying, teach me how to do
Wanna do the dance for it?
You've been trying. You've been paying attention? Here we go.
Thank you.
Oh, Jacob. All right. So you already have part of what you need. you have two sets of drawings, right? One for photosynthesis, one for respiration. And so you have the drawings and you have blanks of the drawings, right? Okay. You got to be able to fill in those blanks. Practice that. And then I just handed you this. This just gives me so many options for testing. I didn't want to give you anything that I hadn't given you. So I'd be ready to fill something like this out exactly. I'd be ready to be asked individual questions. So for instance, look at ETC. I could just based off of this, I could say, hey, where in a chloroplast would I find the electron transport chain? And you would say? Thylakoid. And if I said, where in respiration would I find an electron transport chain? You would say? On the inner membrane. I could also very easily ask you, I think very fairly go, which organelles, plural, have an electron transport chain? And you would say? You don't know where a thylakoid and an inner membrane is, huh? Those are in chloroplasts and mitochondria. Then you look at, well, what does the electron transport chain do? Remember, you've got electrons moving, right? So it's using the energy of these electrons. Along the way, as the electron moves, something happens. What happens? It gets pumped, right? Hydronium ions, H+ ions get pumped. Creates a concentration gradient. That concentration gradient is used to power ATP synthase, which makes ATP. And that's why I wrote down here, use this energy of electrons to generate H+ gradients to make ATP. And then this final question, well, which process does this happen in? Well, it's in both. You'll find this for photosynthesis, you'll find this for respiration. NADH and NADPH, they sound alike, but they are not the same. They have similar functions, but they're not the same molecule, right? They're both electron carriers. Just remember that the P goes with photosynthesis, okay? That's really what you need to know. So they're both carrying electrons, one in respiration, one in photosynthesis. NAD plus and NADP plus, these are carrier molecules. These just don't have the electron yet, right? Okay, they accept the electron, but they don't have one yet. FADH and FAD plus, these function very similarly to NADH and NADPH. They're found only in respiration. I'm not making you know that, oh, they're found in the Krebs cycle, though I could, but I'm not, okay? So you're welcome for that little tiny respite. Yeah, they're found only in respiration. You do need to know that. The capturing of CO2. Okay, so something I don't have on here, but I've told you so many times that you must know it, and you must. What's my formula for photosynthesis?
C6H2, I don't know.
Okay, I'll go with that. I'll write C6H12O6.
Plus 6O2. Wait, do you want the full thing?
Yeah, give it to me.
Plus 6O2 plus 6CO2 plus 6H2O.
Okay, 6. You said it funny, but you got it.
Well, I didn't know you wanted. I could have said
It's
it that way,
good,
but
it's
I
good.
said it. Okay, yeah, okay.
I'm gonna take it. I'm gonna take it.
I know what
6CO2.
you meant. I know what you meant, though.
Oh, this is wrong. Here, let's do this.
Yeah, I know what you meant.
6CO2 plus 6H2O yields C6H12O6 plus 6O2, right?
Are those flipped, the 6O2 and the C6H12O6? Does that go last?
But it doesn't.
Doesn't matter?
No, as long as they're on the right side. Okay? So, of course, this way, right, this is what process? Photosynthesis. This way is respiration. They're inverses of each other, chemically speaking. Okay. Formulaically, so that's good. All right. Why did I have it? Well, because I want you to see what carbon dioxide capture happens, in photosynthesis. Right, so we need to know that. Well, Where specifically does it happen? You do need to know that it happens in the dark reactions. Did I write that down on here? Do me a favor, there at the far, far right, put down dark reactions next to photosynthesis, okay? You need to know that specifically that it's the dark reactions. I guess you can put it wherever you want, just so that you're like, okay, I really, really know this. CO2 capture happens in the stroma, but it happens in the stage called the dark reactions. Well, why do we capture CO2? It gives us some, remember, in anything, where do I find the energy? It's in the bonds. I gotta have bonds to store the energy. So we're harvesting the energy of light in photosynthesis. We need to make bonds to store it in. And so that's, the CO2 is giving us a medium to actually store the energy in. And so that's what's happening there. It's part of photosynthesis. CO2 production, right? That's the other side. That's when we're doing respiration. It happens intermembrane space, the mitochondria. Okay. Allows energy to be released from bonds. So one, we're taking CO2 and we're putting them together and storing it. The other one, we're breaking it apart and we're releasing it. And it happens in respiration. The next one, O2 splitting. Okay, so remember we're splitting an O2 and then we're combining it with some hydronium ions and some electrons. Where does that happen?
At the end,
At the end of
the
respiration. Good, brother. Very good. Okay, so it's part of the electron transport chain of respiration. It's exactly what you just said. It allows the electrons to be stored in water in respiration. Okay, and then O2 formation is the opposite, right? This is a product of the electron transport chain of photosynthesis. It's simply a byproduct of photosynthesis. Useful for us. Doesn't do anything for the plant, though. Photosystem 1 and photosystem 2, they're both part of the electron transport chain of photosynthesis. Remember that the first one in the chain is which one?
Second.
Photosystem 2. It's just a coincidence, just one of those things. They found the first one first, but they found the one that actually happens second. All right, so you know about that. Chloroplasts. What does a chloroplast do? Chloro, think of that word. Chlorophyll, chloroplast, what do they do? Photosynthesis, right? So chloroplast is the specific membrane-bound organelle of photosynthesis. Let me just real quick.
chlorophyll, yeah, it absorbs the light, right?
Chlorophyll does.
Chlorophyll.
So I'm going to draw a chloroplast, okay? Kind of looks like this. And these are all green, as somebody was saying, because they have chlorophyll, right? So this is a chloroplast. This is the membrane. If you don't know this, I draw it, okay? This is the membrane. It's called a thylakoid. The space inside the thylakoid is called the lumen. The space between the thylakoids and this other outer membrane is called the stroma. Okay, it's all liquid. When we said earlier, the dark reactions happen in the stroma, it's saying they happen out here, not in here, not on here, but they happen out here. The electron transport chain and stuff, that happens here, on these thylakoid membranes. Okay? Your chlorophyll, if you're like, well, where exactly would I find chlorophyll on this thing? It's going to be all along here. Okay? All right. So that's a one thing thing to keep in mind is it's a double membrane system. You've got an outer membrane here, and then you've got membranes here, which means I can have one concentration out here in my wow. It's like when you're on the computer and it just goes waiting. My brain's just not working. Your cytoplasm. You have one concentration stuff in your cytoplasm, another one in your stroma, and another one in your thylakoids. So all this compartmentalization is really helpful and necessary for our concentration gradients for the electron transport chain. All right, chlorophyll. It absorbs the photons of light. It's part of photosynthesis. Mitochondria. This is part of respiration. So here's chloroplast. Draw mitochondria. It is also a double membrane system, right? So I've got my outer membrane. I have my inner membrane. I have my matrix. And then I have my that means between, inter-membrane space. The same thing that this did with having two membranes, let me have a different concentration here, here, and here. We have that here. We can have a different one here, here, and here. My electron transport chain is actually on this membrane. In both of these, the electron transport chain is on whatever the inside membrane is, whether it's called an inner membrane or whether it's called a thylakoid membrane, that's where it's at, okay? All right. What does the mitochondria do?
Everything happens.
Say it. Respiration. Very good. Okay. Matrix, that is found in the mitochondria, and it's the innermost part. Lumen, that's the inside of the thylakoid of chlorophyll. Or that should say of a chloroplast. I had a typo there. Sorry about that. Stroma, that's the space between the thylakoid and the chloroplast membrane in the chloroplast. ATP synthase. Where is it? Well, we'll find it on either one of these. It's part of the electron transport chain. Right, and it's going to make ATP from ADP and PI. What powers ATP synthase?
Is it the engine?
The hydrogen ions, which are formed by the pumping and the concentration gradient and all that kind of stuff, right? 3 of them pass through, you make 1 ATP. All right, it's found in both photosynthesis and respiration. Krebs cycle. This is in respiration. I'll find it inside the matrix of the mitochondria. And it splits those two pyruvates into and it releasing CO2, and it makes NADH and FADH and ATP. Glucose production happens in the dark reactions in the stroma of the chloroplast. Stores energy of light as chemical energy. Glycolysis. This is kind of the opposite of glucose production. This is glucose splitting, right? And where does it happen? Do you remember?
Outside.
Outside, in the cytoplasm. Good. It's a splitting of glucose. It produces 2 ATP. Pyruvates. They contain energy in their bonds. They're what's just produced by the glycolysis found in respiration. And of course pumps. We'll find pumps in both electron transport chains, both on the inner membranes. They're going to convert the energy of electrons flowing through. They're going to pump it to make a concentration gradient to create the potential energy for ATP synthase to make ATP. And you'll find it in photosynthesis and in respiration. Yes, sir?
So right here on these dashes, we don't get it right here?
Yep, no, there's nothing to write there. Okay. So I've given you one of these filled out. I've given you one of these blank so that you can practice. All right. Okay. You got any questions for me?
This is gonna be on test. Thursday?
Yeah,
So it's
just, I'm gonna look at this and the drawings, and that's what I'm gonna make the test off of.
So he's got,
If you know this, you're gonna be in a great way.
is there gonna be a lot of questions, like no multiple choice?
No multiple choice.
So just the drawings and.
Yeah. Remember last time, I said, hey, it's gonna look like the homework. And it looked just like the homework.
Oh, yeah.
But I don't know that I was taken seriously. Everything okay? You pray for anything? All right. Any questions? Otherwise, we're gonna be done with this. All right, so I'm gonna erase the board and we'll get started with DNA. If there's a concept you're struggling with, remember, lectures are up online, any video I've ever shown you is up online. Now, where were we with DNA? Where did we leave off?
I don't know, is that on Thursday?
RNA change, that's the last thing I wrote.
Oh,
Did I do that Thursday?
Three types of RNA?
No, I have a, yeah, it's RNA primer, RNA,
so we're looking at DNA replication or RNA
lagging,
transcription?
lagging,
Lagging strand, leading strand, okay. All right. I know that we have done transcription as well, right? How RNA is made, mRNA is made, and we looked at translation, how it's turned from one thing to another. You should have some sheets, right? Get those sheets out real quick. Should have something that looks like this. Let's just do a very quick review. I'm going to give you original DNA. You're going to give me the complementary DNA. You're going to give me the mRNA. You're going to give me the tRNA. And you're going to give me the amino acid sequence. Okay. You ready?
I don't have those papers.
You know? Okay. Let me give you this one in just a second. Let's go. You might want to write this down.
So this isn't the sheet you gave us.
Hmm?
This isn't on the sheet you gave us.
No, I'm asking you to write this down right now.
oh, can I also have one of those fake?
I got one. No, no I would expect something like this on a test, one form or another, when the time comes. All right, so if my original DNA is TAC, what's the complementary DNA? What is it? A, T, G. Good. What would my mRNA be?
UAC.
What is it?
UAC.
UAC?
What'd you say? That,
I want to know what this would be.
U,
I think you got it backwards.
T,
No?
A,
So-
U,
Both the
oh,
complementary,
original, okay, I was looking at the.
both the complementary and the mRNA are
Okay, yeah.
are directly produced from the original. They're going to correspond to it. So if the original is T, then the complementary will be A. If the original is T, then the mRNA will be A. If this was an A, then this has to be T. If this is an A, then this has to be
U.
U. C goes to G, C goes to G, right? If this is my mRNA codon, then I want to know what my tRNA anticodon must be. Remember, these two have the pair, so this has to be
U,
U,
A,
A, C. Good. And now you look on your sheet and you find, okay, and that's coded for mRNA. So if my mRNA code is AUG, what is my amino acid?
MBT.
Is it not the start codon?
Which one is it? UAC?
Give me 1 second.
Not wrong.
Tyrosine. So this one's methionine. Okay? Now, this next one, my tRNA codon is AUG. So what would my mRNA codon have been?
U,
UAC.
C.
UAC, good. What DNA gave me AUC? Or UAC rather?
T, A, C.
tRNA?
TAC.
A pairs with T.
tRNA.
No, you.
No, wait, I thought you said what gave you like UAG.
AUG would have paired with UAC, right? Yes? Because this is an anticodon, it pairs with this codon. Kevin, let me give it to you another way. I got this tRNA thing happening here, okay? And it said AUG. So I want to know what sequence. I want to know what sequence of mRNA this would have paired with, right? A pairs with U, U pairs with A, and
C.
C. Now this is my mRNA. So now I'm going, well, what sequence of DNA would have given me UAC? Okay, A pairs with T, C pairs with G. Remember, there's no T's in mRNA, but it's equivalent of a DNA T, right? And so T pairs with A. So this would have been ATG. So I go up here and I go, all right, that's ATG. And now I want to know what complementary DNA this would have been. A goes with
T.
T goes with
A.
and G goes with C. And now I go, okay, UAC, look on the sheet, what does that code with?
Tyrosine.
Tyrosine? Tyrone, whoever? Alright,
Yeah.
y'all are a mess.
That's
Alright,
how I'm gonna remember every time.
Oh, very good. Whatever it takes. So now I'm giving my complementary DNA. I want to know what my original DNA would have been to give me TCG.
AGC.
A what?
AGC. GC.
Okay. But you know it. What you saying? AGC. So if the original is AGC, what's the mRNA?
U,
U,
CG.
C, G. Good. What would my tRNA have been?
A,
A?
C,
No,
oh,
ma'am.
no, sorry, A, G,
Uh-huh.
C.
Good. And now UCG, my mRNA codon, what does that code for over here?
Leucine.
UCG.
Siren G.
Serine. Yeah.
Siren.
Okay, good.
Not siren.
Okay. Now, I want to draw your attention to something that I've drawn your attention to before. Notice that these are the same, right? The only difference here is T's become U's. The complementary DNA and the mRNA are at least like first cousins here because they're both coming off of the original DNA. All right, so that's a quick check. They should be almost identical. And since these both pair up with these, my tRNA is going to be almost identical to my DNA, my original DNA. Okay? So when you do this, that's a simple check. That's how it should work out. And of course, my amino acid is going to come off of my mRNA codons. How many bases in a codon?
3.
3. Every 3 bases is a codon. You're thinking of something else. We'll get there in a second. How many bases in an anticodon?
3.
3. How many codons are there?
Oh.
How many?
Like that.
64.
Oh.
64. How many anticodons are there?
64.
64. How many biologically active amino acids are there?
20.
20. Use this word, ready? Redundant. There's redundancy built into the system. So take a look on there. How many different codons code for methionine?
1.
Is it only one?
Yeah.
Okay. How many for serine?
4.
Hmm?
4.
4. How many?
Oh, wait, no. 4. No, because it's more than
6.
one spot.
Is it 6?
Yeah.
Okay, good. See, here's one thing that's interesting. It would be possible to change one base in your DNA, potentially, and not have any effect at all. Because there's redundancy in the system. Because more than one sequence of DNA will code for the exact same amino acid. Okay? So it is possible to make certain changes to your DNA and have it have no effect whatsoever on anything. This is called a silent mutation. Write that down. We'll teach you about some DNA mutations real quick. A mutation is any change to the DNA, okay? Okay, the first mutation, I realize that I started with a silent mutation, but I need to first talk about the parent mutation. It's called a substitution. So, a substitution is any time that I swap out one base for another. It would happen accidentally. Remember, there's base pairing specificity, A2T and C3G. That way you always get 3 rings across the middle, right? Because you have things that have 2 rings, and you have things that have 1 ring, right? And so you should never end up with 4, and you should never end up with 2. And also, A only pairs with T, right? They form 2 hydrogen bonds. A does not bond with C because they don't form hydrogen bonds, but they would fit. They would fit together. So it's possible that you could get a substitution. If we accidentally put the wrong thing in, and the little enzymes that are going along to proofread your DNA don't catch it. That's possible. Okay? That's a substitution. We're swapping one base for another. Fair enough? Okay? Substitution. This can go one of 2 ways, okay? One is that we have what's called a silent mutation. And in a silent mutation, it's going to end up, it's still going to code for the same amino acid. This we know is because there is redundancy in the system. We could also end up with a beneficial mutation. Maybe we just picked up a new trait that we didn't have before. Maybe all of a sudden you can roll your tongue, and you couldn't do that before. Well, that's cool. Okay? And maybe we got a deleterious mutation. This is harmful. Okay? Deleterious mutations are harmful. This is potentially something that can kill you, or at least make your life very, very difficult. Sickle cell anemia we talked about. How it's a single change to a single base which changes only one amino acid, but it makes it so that your hemoglobin and your red blood cells, they at altitude, they stick together. They form these weird shapes that they wouldn't otherwise. And so that can be very difficult. It can be life-threatening. But and also, so it's deleterious in one way, but it can also be beneficial. You're far less susceptible to malaria. And so, you know, you might even think to yourself, well, why is it, so here's something that's bad, like sickle cell anemia. If it kills people, why aren't they all dead? Why does it keep getting passed on? Well, one of the reasons that it keeps getting passed on is at certain times it can be highly beneficial. If you have a village and you got a couple people with sickle cell anemia and everybody else doesn't, and there's a major outbreak of malaria, then maybe your people with malaria get killed and your people with sickle cell anemia survive. And when when the genes repopulate, well, whose gene pool is there to repopulate with? It's the ones who have sickle cell anemia. And so it can get passed down that way because some environmental factor, whatever, just wiped out the competition, and all of a sudden it was beneficial for them. Just so you know, that principle that I just described there, that is one of the foundations, something that we do see happening like that is one of the foundations for the world system of evolution. Okay, they would say, well, this happens over and over time. There's selection, there's natural selection, there's environmental factors that suddenly make the environment favorable for this trait that otherwise might have not been. And so it selects for that trait. Everything else gets wiped out. These things become dominant and they fill the gene pool. Okay? And the thinking would be, well, if this happens often enough, over enough time, well then we'll only be left with you know, the different things that we see. Want everybody with me here, okay? Put everything down. Everything down. So, I want you with me. When people talk about evolution, okay, they're saying that in natural environmental factors interacting with a genetic pool. Literally with the A's, the T's, the C's, the G's, okay? Now, it's not actually coming in and going, oh, let's have this T, let's have this A, let's have this G. No. But A's, T's, C's, and G's turn into RNA, turns into protein. Protein determine your characteristics. You have red hair because of proteins, because of substitutions. And so your hair's red, not brown, not blonde. Somebody's hair's curly and not straight because of genetic mutations. Somebody's skin color, they have more melanin versus somebody else because of traits. Somebody's taller than somebody else because of genetic traits, because of mutations, slight alterations to the DNA. Right? We've heard this term of a gene before. A gene is the information that codes for one protein. Your genes have to be expressed in the form of proteins. And then protein, this gene expression, these proteins, the environment can interact with. We see that, okay? We see it all over the place. You understand, like if we're gonna pick people for a basketball team, well, what traits do I want to pick for? Tall. What else? Hand-eye coordination.
Fast.
Speed. Right. Okay. Does white or black matter, or brown, or whatever, red, orange, yellow, whatever colors, does that matter? No, but if you're fast and you're tall and you're coordinated, it's a pretty good set of traits, right? Okay. If there were a famine, so there's just not a lot of food, do I really want to be tall? Do I want to be big? Think about football. Like, what are some good genes, good traits for football?
Strong.
You want to be strong. You want to be big. You want to be, you want to, an NFL lineman. Like, what is that guy? He's 6'4", 300 pounds. Tough to be him in a famine, though, isn't it? He's not getting enough food. In fact, if it gets bad enough, he might be food. Y'all didn't get that joke. Okay. So it's the idea that different traits confer an advantage at different environments, right? And the belief being essentially that the environment changes in the environment would select one. What makes it favorable or not favorable would be the conditions in the environment, and they're going to select for one over the other. And so they're going to, all of a sudden, one trait in the pool of all the traits in a population will be amplified because they're reproducing more and the competition is being wiped out, and so it changes the makeup of the gene pool. Again, thinking that over enough time, if you had enough of these, you get different species. We definitely see genetic mutations. We definitely see gene pools. We definitely see things in the environment. There's no Christian should even begin to say that these things are not true. It comes down to the question of speciation. It comes down to things like, well, okay, what is necessary for life? What do you have to have for life? You've got to be able, at the very least, you already know right now, you've got to be able to do glycolysis. You must be able to split sugars into pyruvates and release ATP. If you can't do that, you can't live. But you've got to have the perfect enzyme to do that. If I don't have that exact enzyme, it can't happen. If I don't have that enzyme, I don't have life. There's no time for selection. You either have it or you don't. You know what I mean? There's you have it or you don't. So before that thing existed, there was not life. You think about the double membrane systems of some of these organelles. They either have that or they don't. You either can handle oxygen, some things can't handle oxygen. They cannot live around oxygen. You can either handle oxygen or you can't. Oxygen either kills you or it's highly beneficial to you. There's no middle ground, which means you had to have all the right enzymes. You couldn't experiment. Oh, can't handle oxygen yet, let me dive back into it. No, you can either deal with it or you can't. It's either an advantage or it's not. There's so many things in nature. There's no room for evolution to be possible because it's an all or nothing proposition. Think about something like an eyeball. As complex and amazing as an eyeball is, it either works or it doesn't, right? There's no time. It works or it doesn't. You either have all the parts and pieces or you don't. So again, we see mutations. They're an important component of biology, right? The environment can select for them or select against them. This happens, there's no denying. No Christian should be trying to deny it. No one should be trying to deny it. It comes down to, well, how do we get species? Okay? All right. So mutations. Any change to DNA. Particular type, the first type, substitution. Swapping one base out for another. It can be silent, where it produces the same amino acid. You would never know that this mutation happened. It can be beneficial or it can be deleterious. This is environmentally specific. Okay? Then we have insertions. Actually, I'm going to go frame shifts. Let's go. So we had substitutions, now let's do frame shifts. So in under a frame shift, we're going to have a substitution, or I'm sorry, we're going to have an insertion and a deletion. Insertion and deletion are exactly what they sound like. The question what is a frame shift? So while you're doing that, I'm going to erase this other side, and I'm going to draw a sequence of DNA 3 times. And we're going to look at 2 types of frame shift mutations.
I got a question. I think
Please.
it goes back to this.
Tell me.
It's a little off topic, not really.
It's okay.
But do you think like humans came from gorillas?
No.
Or some type of monkey? What do you think?
I think God literally on the 6th day made Adam.
My dad thinks we came from monkeys.
So that's it.
Yeah.
I just thought we like transferring off cavemen and then
Well, that's
slowly.
it's what, over the last 100 and some years, scientists have tried to say, you get Charles Darwin, right? He's famous for his discovery of a system, a theory of evolution. How do we've known that there's variation in traits for a long time. We knew that. We didn't know like a genetic basis for it. Darwin did not know about DNA. Okay? He just said, hey, there's these traits. He famously goes on a trip on the HMS Beagle where he's the ship's naturalist. He wasn't trying to discover evolution or heresy or anything else. He just was just going to check out what there was to be seen. Collect specimens, notes, report back, whatever. So they go to the Galapagos Islands. You've got all these different islands. They're close together, but they're separated. So you've got, maybe, we'll go with this finches. So you've got these birds on these different islands. They're all related, okay? They're all finches. They all have most of their characteristics the same. But they have different traits. Some of them have like really, really hard, thick bills. Some of them have very long, slender, needle-like bills. Some of them have bills in between. Well, he's doing observations. He goes, hey, these ones with these really hard, thick bills, they seem to live in an area where there's a lot of, like, I guess he's watching, they're crunching nuts and stuff. And so you think about that. This hard, thick bill is really good for crunching nuts. You know who doesn't live on that same island? The ones with the really thin slender bills. A really thin slender bill is not very good at crunching nuts, right? Now, these two birds are related. They can interbreed, they can produce offspring with medium-sized bills or whatever, okay? They're not different species, but they have really different characteristics that are really well adapted or suited to different environments. The ones with the big beaks live where there's on an island where there's lots of nuts. The other one, they seem to live in an area where they're eating like little insects and stuff, maybe getting them out of trees or getting things out of flowers and stuff like that. And they're really well adapted to that. So we would say, well, we've got variation in a gene pool. He would say you have different traits. And he would say that the environment has selected for these different traits. Okay? But they're all related. These can all interbreed. I do not believe in evolution, but I believe in what he saw. I believe that this one's well adapted for this, and this one's well adapted for this, and this one's well adapted for that. And but they're all the same species. His thinking is if you go far enough into the future, if you could isolate these islands so that they didn't interbreed anymore. Okay? So I just had this area with only this type of bird, and only this type of bird, and only this type of bird. And if you got enough additional mutations, at some point, they couldn't interbreed anymore. A horse and a donkey and a mule. Do you know the relationship between a horse, a donkey, and a mule?
No, it's not related to mule, and mule can't reproduce.
Yeah. So a horse can reproduce, a mule can reproduce. A horse and a mule can reproduce together. But when you breed two horses, you get a horse, and a horse can make more horses. You get two donkeys, they can make a donkey, and the donkey can make more donkeys. But you get a horse and a donkey, they make a mule, and a mule can't reproduce. So evolutionists would say, well, this is an example of evolution in the happening. These two are so closely related still that they can reproduce, but if they reproduce, it's sterile. And so we are just at this moment where we're watching speciation happen. Maybe. I don't think so, but if I'm going to be fair, I'm going to say, well, okay, well, this is where that's at. This is what they're talking about there. Why was I talking about that at all? It happens also. Lions and tigers, they can interbreed. They can make a liger. A zebra and a donkey can interbreed. They call it a zonkey. What a fun name is that? A zebra and a horse. You can Google this stuff. A zebra and a horse make a zorse. These are real things.
A Zorse?
A zorse. If it's anything with a zebra, you got to get that Z in there, right? A zonkey, a zorse. So, fun times. What'd you look at?
The liger, I didn't see the
Yeah.
liger, I see that one.
Yeah, those are cool. So, well, first of all, speciation would require, according to evolutionary biologists, would require millions and billions of years. Okay. You would still, which we've never seen speciation. Okay, never seen it. Then you'd also have the question of how do you get the first thing? If you have to have just the right enzymes so that you can do glycolysis, where did that first enzyme come from? Like, it either works or it doesn't. There's no like, oh, it will make one ATP. No, it either makes two ATP or it does not. So life requires energy, right? So we can either make this energy or we can't. And never mind that there had to be a plant to make the stuff, the glucose. Like, who made the glucose? Where'd that come from? So just even the start, where did everything come from, is an insurmountable obstacle for evolution. There's no answer for that. So I would begin there with where to think about. Like, how could that even be? Biologically, it's impossible. In terms of physics, it's impossible. Everything in the entire universe goes from order to disorder. Remember me talking about that? You're an anti-entropy machine. Life is literally impossible, and yet here it is. But by the laws of physics and thermodynamics, it can't happen. But we see it everywhere, except only here. We don't see it anywhere else in the universe, only here. So when God says you're fearfully and wonderfully made, I believe that. I absolutely believe that. You think about how unique mankind is compared to every other animal, right? I mean, we're the only ones with consciences, right? We're the only ones that can make moral decisions. We're the only ones that can choose right from wrong. We are unique compared to everything else that exists. Well, the Bible says that. It says that of all the things that God made, only mankind was made in his image. That's a complicated concept, but we know this: God is the most moral being. God perfectly chooses right from wrong. We have that capacity as image bearers, very differently than a monkey, than a source, than anything else. You all right over there? All right. So, it's a good question. Any other questions related to this right now? Any other thoughts? You have to put your faith in something. I think you do anyways. You're either going to trust a theory with giant holes. Where did all this come from? And you're just going to, and you're going to go, okay, well, I know that it can't answer it. I know it's not telling me the answer, but I'm just going to choose to believe it. Or you're going to believe in some religious system. And you're going to have to go, well, what's my evidence, right? I mean, blind faith is worthless, isn't it? Trust me, I don't believe in the Bible just because I picked it. I've known people like this. They join a church. I'm going to be Catholic, I'm going to be Mormon, I'm going to be Buddhist, because they just picked one. Blindly. To me, that's foolish. So why do I believe the Bible? Do you believe the Bible? What do y'all think? Why do you believe the Bible?
Believe it.
Is it just blind faith? Why do you believe it?
I wouldn't say it shows proof, but it shows somewhat of evidence.
Okay.
Not that you can prove anything though. So.
Okay. What else?
Well, as literature, it's one of, it's the most historically proven book ever.
Tell me about that.
Well, the amount of documents that are found, and the amount of cross-references that you have and similarities, compared to any other book, especially old book, it's just incomparable. I think, I don't know where I heard this, but I believe that it's more, because we have more evidence that Jesus lived than we have Shakespeare living. Literally, for literally.
Absolutely. Let me tell you how amazing the Bible is. Do it fair. Just stay with me. Nothing else going on, okay? Why do I believe the Bible is true? Book of Isaiah. In the book of Isaiah, we see several things. One, he says, hey, the northern kingdoms, you're going to be taken captive by Assyria. Southern kingdoms, you're going to be taken captive. I don't believe that he names them. He may. I can't remember if he names them by Babylon or not. He does say that then you're going to be set free by a man named Cyrus. Okay? This is prophecy, right? Calling your shots. I'm Isaiah, God speaking through Isaiah. Northern kingdoms, you're going to be taken captive by Assyria. Adios. No return mentioned. Southern kingdoms, you're going to be taken captive. You're going to be set free by a specific man named Cyrus. Okay, that's weird. Then you go to Jeremiah. Prophet Jeremiah, about 100 years later, prophesying just before and then during and then a little bit after the fall of Jerusalem to Babylon. Okay? He tells them, you're going to be taken captive and you're going to remain captive for 70 years. 70, like that's super specific. 70 years exactly. Then comes Daniel. Daniel's a prophet during the Babylonian captivity. And Daniel says, hey, it's been 70 years. God set us free. And then you go to the book of Ezra. And you see that the Babylonians have been defeated. Actually, that happens during Daniel, by the Medes and the Persians. And there's a new king. And his name is Cyrus. And he sets them free. Now, this all happened. Israel definitely was taken captive in Babylon. Israel was definitely set free. The captivity happened in 3 waves. The freedom happened in 3 waves. And the cycle between the first captivity and the first freedom is 70 years. And the second captivity and the second freedom is 70 years. And the third captivity and the third freedom is 70 years. Those things happened. At first they tried to deny that they happened, but then you go to Babylon and you find evidence that Israel was there. And you go to Israel and you find evidence of these names. You find names of kings in the Bible that you don't find anywhere else in history. And so they go, well, see, the Bible's a liar, until they go and find them. And they go, oh, well, holy moly. At the very least, the Bible is a pretty accurate historical document. They tried to just discredit it, but can't. So then they go, okay, well, prophecy's not possible because there is no God. Or prophecy's not possible because if there is a God, he doesn't actually interact in human events and tell people what he's going to do. So Isaiah's got to be lying. Isaiah couldn't possibly have been written by the original Isaiah. Somebody had to have written this after the fact. Except you find manuscripts that are too old for that to be the truth. So if the Bible is historically accurate and all this happened, and Isaiah prophesied about it hundreds of years before and names the guy who sets them free, that's some evidence I got to really deal with, don't I? That's incredible. There have been a lot of prophets. Think about people like Nostradamus. At best, their prophecies are vague, and almost none of them ever happen. Or we have to really squint to think. But God says Cyrus is going to set you free. I'm going to move in his heart. And he says a lot more than that also, by the way. He says, hey, I'm going to use this people to pay back this people. And he uses the Babylonians to pay back the Assyrians. He uses the Medo-Persians to pay back the Babylonians. He calls it with such incredible detail that you can't deny it as history. And then you can't even deny it as an artifact. So then you're just left going, well, that's true. And if that's true, then who is this God who revealed himself and who revealed his will and his plan for his people and for the future and for all of creation? And what else does he have to say? Because this is only like one little piece in a very big story that's about salvation. From Genesis chapter 3, where we see the first sin, to Revelation, it's all one story about how God is going to redeem the world and the sin through Jesus Christ. That's something we have to wrestle with. If people will actually look at this. This is one little piece. And it's amazing how all of the Bible points to Jesus. I mean, all of it. What time is it?
44.
That was a worthwhile little excursion, I think. You should go look some of this up. Be sure that I'm not just making it up. It is incredible. Incredible. All right. You got any questions for me? Let me go back to where I was at. You're going to put your faith in something. Don't put it blindly. Let there be evidence for whatever it is. Don't be fools. Let there be evidence for whatever you're going to believe. If you're going to believe in God, then have some evidence for it. If you're going to believe in evolution, then find some evidence for it and figure out a way to disprove that. They've been trying for hundreds of years. There's an old expression. How do you defend the Bible? Because the Bible's been attacked for hundreds of years, ever since the Age of Enlightenment in the 1700s. People have just been attacking the Bible, attacking the Bible, attacking the Bible. Well, it's like saying, how do you defend a lion? So how do you defend a lion? You get out of the way. You don't defend the Word of God, you just get out of the way. He will defend himself. All right. You have a test next class. Study. The Lord be with you.
Thank you.